Hi, I'm Ellen Winesapple, a product manager at EpiCypher. Welcome back to our video series on the basics of chromatin mapping. In this video, I'll review how cut-and-run and cut-and-tag work and ideal applications of each assay. So as I said in a previous video, Katana cut-and-run and cut-and-tag assays provide multiple benefits compared to ChIP-seq. But how is this possible? EpiCypher Katana assays leverage recent advances in immunotethering technology, using enzymes to selectively cleave and isolate antibody-bound chromatin from intact cells or nuclei. These workflows skip the most challenging steps of ChIP-seq, including cross-linking, chromatin fragmentation, and antibody pull-down, enabling higher throughput. Less off-target material is captured versus ChIP-seq, generating higher resolution data with fewer cells and sequencing reads. But which assay should you choose? For the rest of the talk, I will review the cut-and-run and cut-and-tag workflows and share EpiCypher's recommendations for assay selection. In general, EpiCypher suggests that most users try cut-and-run for their experiments. It is robust for nearly all targets and cell types we have tested, and the protocol is very straightforward. We offer kits for the assay and for cut-and-run library prep, making it easy to set up the entire workflow end-to-end in your lab. Let's review the basic steps of cut-and-run assays. To start the assay, isolate cells and immobilize them on magnetic beads. The use of a solid support makes it easy to wash cells and process reactions in 8-strip tubes. Cells are permeabilized and treated with an antibody to the target. You then add a fusion of protein A, protein G, and micrococcal nuclease. The protein AG binds to immunoglobulins on antibodies, tethering MNase to antibody-bound chromatin. Calcium is added to activate the MNase, and MNase clips adjacent chromatin. Cells and off-target chromatin are easily removed with magnetic beads, while clipped fragments diffuse out of cells and remain in solution. Target-associated DNA is purified and prepared for sequencing. Each library contains a unique pair of barcodes to distinguish reactions during data analysis. Sequencing data are analyzed using approaches similar to ChIP-seq, as discussed in a previous video. But cut-and-run only uses a fraction of the sequencing reads and also has improved signal over background. The proof is in the data, as EpiCypher cut-and-run assays produce high-quality profiles for many target classes, including proteins that have been challenging to map with ChIP-seq, such as chromatin remodeling complexes. However, there are certain situations where cut-and-tag can be used. Let's explore this assay. Many scientists prefer cut-and-tag because it is a faster assay and bypasses traditional library prep steps, making it easy to set up large-scale experiments. However, there are some reasons we don't recommend this assay for every project. First, cut-and-tag is not robust for chromatin-associated proteins. Cut-and-tag should generally only be applied for histone PTM targets. In addition, cut-and-tag is more challenging to optimize versus cut-and-run due to high assay sensitivity. This is particularly true for low nuclei numbers or when mapping low abundance targets. Now let's review the main steps of cut-and-tag mapping assays. First, you isolate nuclei, which are preferred for cut-and-tag assays, and immobilize them on beads. You add a primary antibody to the target, followed by a secondary antibody to amplify assay yields. A fusion of protein A, protein G, and transposase TN5 is added to the reaction and binds antibody-labeled chromatin. This is a distinct enzyme from cut-and-run, and the TN5 comes preloaded with sequencing adapters. In cut-and-tag, magnesium is added to activate TN5, and TN5 clips adjacent chromatin and ligates sequencing adapters to DNA. This process is called tagmentation. An SDS buffer is added to release tagmented DNA into solution. And the entire reaction mixture is used to perform PCR. Note that this direct-to-PCR technique is specific for EpiCypher's cut-and-tag assay. Other cut-and-tag methods require DNA purification at this step, adding time to the assay and increasing the risk of sample loss. PCR is performed using primers that recognize ligated adapters, resulting in specific amplification of tagmented DNA. The PCR step also adds barcodes for data analysis. The libraries are purified and sequenced and are analyzed using methods similar to cut and run. If you are interested in learning more or want to see our available kits and reagents, be sure to visit our website. We have nearly everything you need to get started with cut and run and CUT&Tag assays in your lab. If you are interested in other informational videos, let us know. Visit epicypher.com and click Contact Us, or email me at ellen@epicypher.com.